Good morning, everybody. Thank you for attending this webinar for the young epilepsy section. Today, we will talk about methods of EEG recording from standard video EEG to advanced approach. With me here we have Christos Lisgaras from the NYU Langone Health and Nathan Kline Institute for Psychiatric Research and Center for Dementia Research from New York, USA. And if you have questions, you can just write it in the Q&A section. And let's start with our webinar.
Perfect. Thank you. Thank you so much, Bruna, for the kind introduction. And thank you, everyone, for joining the webinar today. So as Bruna mentioned, we will be talking about methods of EEG recording. We will start with some standard approaches, and then we're gonna discuss some more advanced approaches and some applications. So I have nothing to disclose, but first I would like to acknowledge the YES team for putting together this webinar series. It's a great pleasure to be part of the program, and I also would like to thank the ILAE Commission on Neurobiology for their support, and also all the other speakers that devoted their time to give a webinar for YES. So with that, here is the outline of my talk. I'm gonna start with some simple questions. What the EEG tell us, why we need to record the EEG. The focus is gonna be on animal models. How to record the EEG. I'm gonna go over the approaches that people have used. And then I'm gonna transition and talk about how we can combine the EEG with other approaches that are being used in neuroscience research. And last, I'm gonna talk about some applications. So the EEG tell us a lot of information. Some of them have to do about normal brain rhythms. As you all know, the brain can generate various different rhythms that range from very slow oscillations to faster oscillations like gamma. All these frequencies are important because they are involved in memory processing. Some of them are also involved in sleep, and here you can see some of them, like the slow waves, the delta waves, the ripple frequencies, but also some of them, they're also involved in stages of sleep called REM sleep. And this is one example is the theta rhythm. In epilepsy, the EEG is important because we can look at abnormal electrical activity, such as seizures in animals and humans, as well as abnormal electrical activity in between seizures. And for the rest of my talk, we are gonna talk about interictal spikes and high frequency oscillations. So the EEG is really important, not only for looking at normal frequencies, but also for abnormal brain rhythms that are really important in brain disorders like epilepsy, as well as Alzheimer's disease. And I'm gonna be giving you some examples about that by the end of my talk. But why it's really important to record the video EEG? So I'm gonna give you two main reasons. One is that many abnormal EEG activities, even seizures, can be silent or may lack overt behavioral manifestations. So it's not really clear that, for instance, an animal experiences a seizure or has an abnormal EEG because we don't see something weird in their behavior, but there might be some hints that we might have missed, and there may be very subtle changes in behavior. Another reason is that some behaviors may resemble seizure-related behaviors, but may not be actual seizures as defined by the EEG manifestation, the EEG evidence that there is a seizure going on. For more information about that, I'm going to direct you to the reference that I have below. You can get more information. And also by the end of my talk, I'm going to give you some resources, some books that you can read more about that. Electrophysiological recordings can be done at various different levels. So for instance, we can record the EEG from the scalp. We can record the ECG from the surface of the cortex. We can record the local field potential as well as single unit activity. But all of these approaches, they have a trade-off between how invasive we want to be to the brain versus the resolution. So obviously, if we would like to record single unit activity, we need to be very close to the neuron to capture their firing. But if we are more interested in like a global activity, we can either record EEG or the ECG. So there are many different approaches depending on what we would like to look at. So as all of you know, EEG can be applied in humans with many different electrodes on the scalp, or depth electrodes, or electrodes on the surface of the cortex. In animals, it can be done similarly because we can implant mice or rats that model a brain disorder with electrodes, and we can record the EEG in a similar way that the EEG is recording in humans. So essentially, you have the electrodes that are connected to a head stage. Then there is a cable that goes to the amplifiers. We can record it with using a computer. We have a camera that we would like to also look at the behavior of the animals. And then the EEG can be analyzed offline. So similar approaches, largely similar approaches can be used between humans and animals. Here, I'm going to briefly describe what we need to consider when we would like to do a video EEG recording in animals. So one of the first things to consider is to actually determine the areas of interest. Are we interested in recording from the brain surface? Are we interested to go to deeper structures like the hippocampus? Do we like to record even deeper structures like subcortical structures, or we would like to just record like the global activity? So these are important questions that they're gonna guide us which electrodes we're gonna use and which approaches are best for the recording. There are several different approaches. You can use screw electrodes, you can use depth electrodes to reach deeper structures, and largely the video EEG recording is similar, either by using screw electrodes or depth electrodes. So in the next few slides, I'm going to be describing the approaches that we use to record video EEG. The first approach that I'm going to be discussing is the tether recording that involves the use of cables that connect the animal to amplifiers. Some of the advantages of using tethered approaches is that you can record the animal for long periods of time, and they allow often continuous recordings. You can usually customize how many electrodes, how many animals you can record simultaneously. But some of the disadvantages is that movement may be restricted in the animal because there are cables, and they cannot really move throughout all their home gates. But there are several solutions. One of them is to use motorized commutators that allow easier movement of the cables as the animal moves around the gates. But some of the limitations is that when the animals, they have cables, they cannot usually be housed with other animals, because non-implanted mice may compromise the equipment of the tethered animal. So there is a limitation of non-social housing. And that is important because in animal models of epilepsy, people have reported that if animals are being housed alone, they may have increased seizure burden just because they are isolated. So this is something to consider when you are planning your experiments. So the other approach that people have used is called radio telemetry. It's an alternative to the tether recording that includes the use of a transmitter, and it doesn't use cables, in that the animal is freely moving, as you can see it here, and the signal is either transmitted electronically to a computer through a transmitter, or it can be saved in the transmitter, and then you can recover the data. So some of the advantages of the approach is that movement is less restricted compared to tethered. It can be applied to small animals like pups, as you can see here. They can go back to the home cage with their mom. And social housing is possible, as you can see here. But some of the limitations are that the battery of the transmitter is quite limited. You cannot record usually for several weeks. There is a limited recording bandwidth. If you are interested in looking at faster frequencies, that might be a limitation. Also, this approach is usually a less cost-effective approach, and it may be challenging to use the same implants over time in different animals. So these are some things to consider. And again, you can learn more about that in the book Models of Seizures and Epilepsy in a chapter that Ed Bertram wrote. Now I'm gonna talk about more advanced approaches that people use, and some of those approaches involve the use of silicone probes. So these are very thin electrodes that can be implanted in a rat or a mouse. And there are many different electrodes that are spaced very close to each other. And that is advantageous in that it gives you a very nice profile, very detailed profile with high spatial resolution of the different activities that are being generated in different parts of the brain. And you can actually record from different subfields. This is the hippocampus of a rat. This is the dentate gyrus here. This is area CA3, CA2, C1, and subiculum. Depending on your implantation, you can actually record from many different subfields simultaneously. And this is really interesting because there are some activities that are very focal. So if you have electrodes like those, you can actually address whether there are some activities that were missed, and by using just screw electrodes or just a depth electrode. And people have developed different approaches to implant those electrodes. They use micro drives, as you can see here. You can move the electrode up and down depending on where you are planning to record. And then you can actually recover the electrode. You can clean it up, and then you can use it for a new animal. So this is a really nice approach. And again, you can find more information in the references. Another type of electrode that is large scale, it's considered large scale, involves the use of neuropixels. So these are probes that are quite long. As you can see here, the track of the electrode. It has many electrodes, like thousands of electrodes placed very close to each other. And that gives you the opportunity to record from many different areas simultaneously. you can get activities like ripples, units from pyramidal cells in area CA1, and you can also record seizures. So imagine the possibility to record a seizure with thousands of electrodes from an animal model and figure out, is there an area that may be generating these seizures? So these questions can be addressed possibly by using these type of electrodes in animal models like rats or epileptic rats or mice. So in the next few slides, I'm going to be describing how the EEG can be combined with other approaches. So the first approach that I'm going to be talking about is optogenetics. Optogenetics is a combination of genetic and optical methods to achieve gain or loss of function of well-defined events in specific cells of living tissue. So essentially, how it works. So usually it involves the delivery of a viral vector. So we kind of contaminate some cells with a viral vector that carry a protein that is sensitive to light. So those neurons are going to express this protein because they were contaminated by a virus that expresses this protein in the neuron. And then those neurons are very sensitive to light due to the protein. So we express this virus in an area of interest, and then we implant an optical fiber so that we can deliver light stimulation at the area that we are interested in. So that gives us the opportunity to manipulate cell types of interest just by shut them off or activate them. And that can be done with different viral vectors. There is a variety of vectors that can be used that are either excitatory or inhibitory. And people have used that in epilepsy research. And this is a very exciting technique. So one of the applications of this technique that has been done by several people now involves the activation or inhibition of specific cell types to understand what is their contribution to seizure dynamics. So these are two papers that you can take a look. There are many others also, but here you can appreciate that if you control cell types in the thalamus, you can actually interrupt seizures. And this work also looked at cell types in the dentate gyrus to look what is their contribution to seizure dynamics. And here is how that works. So we have the mouse here. You record the EEG using an amplifier. Then you feed this EEG to a computer that runs a programmable processor program. So that program analyzes the EEG in real time, and it tells you when a seizure begins. And as soon as the seizure begins, then the program gives a command to the laser to switch the light on or off. So here is the approach. You have some seizures that the light went on or some seizures that the light did not to have a control. And then based on this approach, you can determine whether switching off or switching off, switching on or switching off the activity of a specific cell type has any effect on interrupting a seizure. So you can take a look at these papers, and there are many others as well to look at the specific approach. Other uses of EEG have involved memory assessments and whether we can manipulate the cell type and improve memory. So this is an example in the normal brain. where the authors looked at area CA1 that generates surf wave ripples. And what they try to do is actually to detect those events in real time. Those events called surf wave ripples, they have been deemed very important for memory. And they thought, what about increasing their length or make them last longer? Would memory be improved or enhanced? And that was what they found, that if you generate extra oscillations like those, the rats had better memory. And of course, they did controls and they shine light at times that those oscillations did not occur and they did not see an effect. And we applied a similar protocol in epileptic mice that have a lot of high frequency oscillations, those events that you can see here. So we detected them in real time and we asked whether memory would be improved. And that's what we found. And you can take a look at the paper on bioRxiv. So if you shorten high frequency oscillations, that is sufficient to improve memory in epileptic mice that they have high frequency oscillations during sleep. So that tells us that high frequency oscillations can impair memory, and if you target them selectively, you can actually improve memory. So I also wanted to discuss that all the previous slides I was talking about the EEG and how important it is to look at seizures, epileptiform activities, use it as a method to improve memory. But there's other approaches like video recordings that you can mine and ask whether there are any hidden patterns that we have missed in the behavior of the animals that can tell us whether they have a response to the drug, whether they are going to become epileptic. So I would like to direct you to this paper that they did video recordings and they tried to figure out whether the animals showed something in their behavior, like patterns that could be promising and could predict response to therapy or which animal may become epileptic. And they came up with a syllabus, as you can see here, that the animals were doing quite repetitively, and they thought that this kind of behavioral patterns may be promising if you look at them very closely. Or you can find out more about this approach in the paper that is published. And in the next few slides, I think we're good with time. I'm going to be talking about two applications of the EEG in animal models of Alzheimer's disease and epilepsy. So what we did here is that we used an electrode that has many contacts. This is called a silicon probe, as I described previously. We implanted them in mice that recapitulate aspects of Alzheimer's disease. And we asked whether we could see something similar we see in epilepsy. And in this case, we were interested in high frequency oscillations. And the reason is that high frequency oscillations were considered a biomarker of epilepsy, but we wanted to test the hypothesis that they also occur in other brain disorders, that epilepsy can appear, but the mice were not epileptic at the time, at least that we recorded HFOs. So here you can see the targeting in the hippocampus. There's some trails of a dye that we use to make sure that the electrode reaches its target. Here we use just one electrode, but we have done recordings with multiple as well. And this is what we found. So as you can see here, we have the targeting in the hippocampus. We targeted the dentate gyrus of the hippocampus. Here is the recording from a control mouse. We can see that there is slow activity during slow wave sleep. that this is a control. We don't see HFO activity in these mice, the controls, but the transgenic mice, they have a mutation that leads them to overexpress beta amyloid sometime in their lives. You can see that there is high frequency oscillations occurring during sleep. You can better see it here. And that was significant in many mice that we recorded. Our controls did not show an effect did not show HFOs, but our transgenic mice, they showed HFOs. And interestingly, those mice, they showed HFOs before neuropathology develops. And that is truly interesting because it may imply that HFOs is a very early abnormality in the brain of those mice. And they may contribute to the pathology. We don't know that yet, but that is an intriguing hypothesis. But nevertheless, it tells us that the EEG can give us early biomarkers. Then we recorded from epileptic mice because, as you know, HFOs are very promising is a very promising biomarker in epilepsy. So we recorded them from mice with an injection of cyanuric acid in their brains to become epileptic, or mice that they were injected with pilocarpine. Those two models, they produce mice with robust convulsive seizures. And we did a simple comparison by comparing the duration of HFOs in Alzheimer's mice and mice with epilepsy. And we couldn't find a difference in their duration. So how long those events lasted were very similar between Alzheimer's disease and epilepsy. And also their spectral frequency was very similar. So as you can see here, the events are around the same spectral frequency. So that tells us that HFOs are in AD and epilepsy are very similar, and it would be hard to know whether this is an HFO from an epileptic animal or an animal with AD if we just look at their duration and spectral frequency. The last example that I would like to discuss here is interictal spikes in Alzheimer's disease. As you know, patients with Alzheimer's disease show epileptiform activity in the EEG, and we wanted to test the same, to look at animals and ask whether we could find an area in the brain of those mice that epileptiform activity may be generated from. So we used those electrodes that I showed you before, and we recorded from many different areas simultaneously, from the cortex, CA1, and the dentate gyrus. And what we found was that interictal spikes were largest in the dentate gyrus. They weren't only largest there, but their sources, if you look at the current source density profile, they were earliest in the dentate gyrus. So that tells you that the dentate gyrus is a hotspot of epileptiform activity. The amplitude was largest, and the sources there were very early compared to other areas, meaning that the dentate gyrus has a large contribution to those events. We also looked at high-frequency oscillations in epileptic mice, and here I'm going to show you some results on how we can manipulate those events and test whether they affect memory. So I'm talking about those events here that they occur during sleep. We used a closed loop protocol like the one that I described before. So essentially, we record the EEG in the animals. We detect HFOs in real time, and every time that an HFO is detected, we shine light to inhibit a brain area of interest and test whether that is sufficient to stop the HFOs from continuing to occur, and whether that has an effect on memory. So here is the experiment that we did. So we had mice during a behavioral task. So this is a social memory task. So we have two mice on the opposite sides of an arena. They are confined in a pencil cup, and we have a subject mouse that we allow them um, to explore those two mice. We measure the times that the mouse explored either the green mouse or the blue mouse here, depicted here. Then we switch the position of those two mice to make sure that they don't have an intrinsic preference to the location or the mouse placed in its location. We measure again. Then we put the animal back to the home cage when they usually sleep. And that time is advantageous because the mice at that time, they try to consolidate what they experienced during those learning trials. And that was the time that HFOs occurred. So we asked whether those HFOs would actually interfere with the ability of the mouse to consolidate what they experienced. And we would test their memory afterwards, after 1 hour of sleep, in a similar trial that we kept one of the mice that they saw before and they know, but then we switched the mouse that we had here with a novel mouse. And as you know, mice, they like novel objects, novel animals to explore. So they would go to this mouse and explore it and say, oh, that's something different from what I saw before. And they spend more time with that mouse. So epileptic mice were impaired, so they couldn't tell whether this was a familiar mouse or that was a novel mouse. So they had an impairment of recalling what they learned. And we thought that might be due to HFOs during sleep. So to test this hypothesis, we inhibited HFOs during sleep. Here is an HFO that was not inhibited, and here is an HFO that was stopped by CA2 inhibition. We reduced the power of HFOs during sleep, and that was sufficient to increase the time that the mice were spending exploring the novel mouse. This is epileptic mice that were impaired. And here you can appreciate the differences. So here are normal mice. They are not epileptic. This is a naive mouse. They prefer the novel mouse because they explore more time, and this is shown by a positive discrimination index. Epileptic mice were impaired. You can see it's negative. They don't show a preference between the familiar and the novel. When we stop HFOs in epileptic mice, this is the closed loop group. The mice, they started to recognize the novel mouse and spend more time with them. But when we use the control intervention that we shine light, but the virus that we used wasn't able to inhibit the area of interest because it was a control virus, we did not see an effect. So that tells you that the memory of the mice that were epileptic, we stopped the HFOs from occurring, continuing to occur, had similar memory to the normal mice. And that is very interesting because the epileptic mice were really impaired in that task. So that tells you again that HFOs contribute to memory deficits, at least in mice, and targeting them selectively at the time that they occur, that might be a way to alleviate social memory deficits in epileptic mice with HFOs. And also we looked at seizures. We could shorten seizures. We could reduce the spikes, interictal spikes by doing closed loop optogenetics. And this is the HFO effect. So if you would like to learn more, you can take a look at the bioarchive with this work. So, and with that, I would like to wrap up by saying that the use of EEG is really important. It can give you answers relevant to big questions in the epilepsy and other fields as well. And for more information, you can take a look at the Models of Seizures and Epilepsy book. There is a second edition of this book, the Jaspers, Basic Mechanisms of Epilepsies. I think there is a new version, the 5th version coming out. For people that are interested in more computational stuff, you can take a look at MATLAB for Neuroscientists. This is some of suggested books that you can take a look. There is also some opportunities to learn more about epilepsy. One of them is the San Servolo course that is being organized for several years by the YES-ILAE Neurobiology Commission. I have been there. It's an excellent course. It's highly recommended. And also the international course on the pathogenesis of epilepsy, another great opportunity for you to learn more about epilepsy. And two more opportunities. If you are interested in high frequency oscillations, we are hosting a workshop at NYU Langone with more than 33 faculty that will be discussing HFOs for 2.5 days. I would like to invite the trainees to apply. We have funding to support your travel, to come and present your work. So, and the deadline is tomorrow. So make sure you register and submit your abstract. This is the QR code. And also another opportunity is the Mechanisms of Epilepsy and Neuronal Synchronization Gordon Conference. This is a very exciting conference that is going to address the transition of a healthy brain to an epileptic one. So I also encourage you to apply to attend and interact with your peers. And I think that's all on my end. I'm gonna be happy to take questions, and thank you again to YES and everyone for the opportunity and for organizing those educational webinars.
Thank you, Christos. A really nice, clear also presentation. We have two questions. Here, I will read the first one. Will the optogenic intervention to seizure be one of the treatment options for human epilepsy? If you know about human study about this, can you tell us?
Yeah, so this is an excellent question, Auto. Optogenetics are very limited in humans. There is, I think, one trial that tested optogenetic stimulation for restoring vision. It's really exciting. It's haven't really applied to brain disorders, but from that trial, I'm hopeful that there would be some ways to do that in humans. It may be that there's some ways to affect deeper structures if you just inject the virus and shine light outside the scalp. But I think the optogenetic interventions in the mouse can give you answers about whether looking at an area in the human brain and target it with electrical stimulation, you may actually affect seizures with for an area that we didn't know it could contribute. So for instance, for CA2 manipulations, we didn't know whether there would be an effect if you just target a very small area. And the beauty of the optogenetics is that it gives you cell type specificity. Then you can go back and test, with electrical stimulation, that is a pattern stimulation that can be inhibitory. Would you be able to stop seizures? Can we move, for instance, the stimulating electrodes in humans a little bit closer to areas that may be actually very efficient in stopping seizures? So this is what it gives you. It gives you answer and directions. So yes, I hope I answered your question. But if not, please comment. All right, I see more questions.
Yes, we have other three questions. One is like a comment. Do you think it would be useful to have, in your opinion, if possible, a sort of manual of EEG recording activity in rodents for animal research? Sometimes it seems there is not enough consensus on what some activities are and what is their meaning, especially when compared to the clinic.
That is an excellent comment. I think it comes from Rosella,
Yeah.
a person with a lot of experience with EEG. So I totally agree, Rosella, that I think it's really important to have a manual of, is that a spike? Is that a seizure? Is that an HFO? Is that an artifact? I think there are some books that people have used in humans, but in animals, I think it's very limited. I think there is a rodent EEG atlas. There is a website on the internet that you can take a look. There's some snapshots of activity, but of course, this is in possibly normal rodents, or that might be different in pilocarpine, intrahippocampal kainate, intramygdala kainate, genetic models, TBI models. So I think there is a lot of uh, you know, um, uh, knowledge gap, let's say. And I think, it's something that needs to be developed. And I'm hoping that in the next few years, we would be able to address that. But great comment.
We have another one technical question for the recording, EEG recording of rats. During the EEG recording, how do you deal with the noises, which are ambient noises or motion artifact of animals? Is it enough to extract the specific frequency waves of interest? And could you tell me the optional parameter for sampling rate of EEG recording high frequency oscillation?
Sure. So with regards to the first part, how you deal with noises, it's true that there's many sources of noise. When you record an animal, it may come from electrical noise from the outlets. It may come from motion artifacts because the animals are moving around, and they can create muscular artifacts in the EEG. So one of the ways to mitigate those is to use a Faraday cage if you have noise from the environment. So essentially, people use some wired selving or Faraday cage material. I think that's how it's possibly called, to seal the area that you are doing the recordings, and then you can ground this area with your recording system. That it reduces the noise, it filters out part of the noise. But the noises that relate to the motion artifacts could be mitigated by making sure that the ground and the reference are placed are very stable and placed very securely on the animal scalp. If there's many if it's not well stabilized, then you would see artifacts in the EEG. And how strong is the implant? It's also really important, because if you have movement of the electrodes and the whole implant when the animal is moving around, then that's why you see something in the EEG that is an artifact. Then the next question is it enough to extract the specific frequency of it with FFT? Not necessarily. The FFT is going to give you a quantitative view of which frequencies are involved. You can use spectral analysis with spectrograms to also look at whether the prominent frequency is present in where it's present in your signal. Extract specific frequency waves, and the optimal parameters for sampling rate of EEG for high-frequency oscillations. So there is a review paper in Epilepsia by Mikey Zymans that they discussed what is the optimal parameters. Usually people use 2 kilohertz for sampling rate. Because you need at least several times above the highest frequency of HFOs to record them. So if you record up to 5, if you are interested in recording HFOs up to 500 hertz, usually people want to sample their EEG with 2 kilohertz, so you have at least 4 times above. And also the other filters that you would like to look at is the low pass and high pass filter so that you capture the frequencies of interest. I hope that answers your question, but feel free to email me if you would like to discuss more.
We have another one from question from Eleni. How long did you notice the high frequency oscillation typically last, and how fast was the closed loop to inhibit them?
Yeah, so let me go back. I think it was so the latency of the closed loop protocol is below 1 millisecond, and the HFOs are okay, it's here. So it's about 20 to 30. They range. So there is a range of how long they last, but typically it's around 20 to 30 milliseconds in the epileptic mice. But of course, you know, there's variability, as you can see here, even in the same animal. So these two pictures are from the same animal. You see an HFO that is 50 milliseconds, and the other one is like 20. So there is some stochasticity of, you know, let's say, how long HFOs last. But overall, we could reduce them by half almost in duration. So the number of cycles was also reduced, and the power of HFOs. I think that was your question, right? How long did you notice typically last? Yeah.
Mm-hmm.
But feel free to email me. I'm happy to discuss. And there is another question from Cecile.
Which material would you say is best when making EEG electrodes in the lab? Also, would you say is the best thickness of the electrode?
That is a hard question, Cecile, because I haven't tested all the different electrodes that they are in the market, but I would say that the electrodes that I tested myself, and they're in the paper, they work well for the purposes that uh, I have tried to I'm working on. So we use stainless steel electrodes for depth electrodes, and the screw electrodes are, I think, also stainless steel, and the best thickness of the electrode. So it was 50 microns, I think, the thickness of the depth electrode. For the silicon probes, it's also 50, if I'm not mistaken. But for the closed loop optogenetics, you also need to consider that a fiber is going to be implanted. So I bundled the fiber with the electrode. But happy to talk offline about you know, what type of electrodes we use and what work for us, depending on what you would like to use.
Okay, I have one more question. Do you know if
Yeah.
there are studies that correlate the effect of drugs, autism medication, and high frequency oscillation?
Yes, there are many. There are many studies that they used levetiracetam or other drugs to test whether that would reduce HFOs. And importantly, I think in one of the webinars, Premex showed that when you use levetiracetam to reduce HFOs that are above 250 hertz, then you don't reduce the ones that are below, like 80 to 200, that are involved in memory. So it doesn't affect the normal frequencies, but the ones that are abnormal, they seem to help. But in humans, there are many studies looking at how the medication affects HFOs, and how, if you reduce the medication, what happens to HFOs, because they may come back. So they may tell you not only when there is an effect of the treatment, but when you stop the treatment, whether what comes back, it can also be looked through the HFO lens.
And in this human study, is there also gender differences for?
So there is no big sex differences reported for HFOs in humans. I don't think that was an extensive study, but in animals, when we look, there is a trend towards more HFOs in the females. However, it's quite variable, because the females can be in different states of the cycle, and that can affect the excitability. So there are some sex differences, yes, but within the female, there might be also an effect of the cycle that is really interesting to look at.
Okay, and study that evaluate the concentration, for example, of estrogen or progestin in the female patients?
I don't think so. I don't think they looked at that. Yeah.
Because maybe it could be related to the hormonal changes in.
Yes,
Okay.
absolutely. Yeah.
Okay, so I think there is no more question.
Yeah.
So maybe we could close the session.
We could wrap up.
Yeah.
Yes.
Thank you again for your presentation, for as well the several questions, and for your availability. And
Yes.
I hope that the webinar will be useful for our the young people that follow today. So thank you again. Thank you for Naoto for organizing it, and for involving also me as moderator.
Yes, thank you, everyone. Thank you so much.
Bye. Bye-bye.
Bye.